Hello, everyone, and welcome to today's webinar. My name is Olga Bodie, and I'm a Syngap parent and part of the Syngap Research Fund team. Our talk or discussion today is an introduction to Autism BrainNet. And I have the pleasure to introduce today's speakers, Dr. David Amaral and Carolyn Hare. Dr. Amaral is the Scientific Director of Autism BrainNet and the Mind Institute Autism Center of Excellence. He is a professor of psychiatry at UC Davis School of Medicine, and he is engaged in interdisciplinary research into the causes and treatment of autism and related neurodevelopmental disorders. He is a nationally and internationally renowned authority in autism. Carolyn Hare is Clinical Director for Autism BrainNet. She began her career in special education and eventually moved toward autism research. She is a certified tissue bank specialist and works directly with donor families. By the end of this presentation, you'll have the opportunity to get the answer to your questions. We'd love to hear from you. Please write your questions in the Q&A below. For those of you just joining us, welcome. And again, our speakers are Dr. David Amaral and Carolyn Hare, and their talk is an introduction to Autism BrainNet. It's now my pleasure to turn things over to Dr. Amaral. Thank you.
Thank you, Olga. Share my screen so we can get started. Does everybody see that okay? Mm-hmm. Good. Thanks, Carolyn. Well, thank you, Olga, and thank you, Syngap Research Foundation Fund, for allowing us to give you an introduction to Autism BrainNet. Here's an overview of what we'd like to do today. First of all, talk a little bit about what is Autism BrainNet. I will try and give you some hints about the importance of brain tissue donation for carrying out research in neurodevelopmental disorders. I'm going to use one example of work that we've done here at the Mind Institute to show you some of the benefits of studying brain tissue. And I'm going to use the example of studying anxiety and autism. And then I'll hand off the presentation to Carolyn, who will walk you through the donation process, as well as the donor family aftercare program, which she has established. And then we'll end and we'll open it up to questions and answers, and we'll hopefully try and answer your questions to the best of our ability. So what is Autism BrainNet? Well, Autism BrainNet works with researchers and communities affected by autism and other neurodevelopmental conditions to develop a sensitive and effective strategy for acquiring postmortem brain tissue. We realize that some of the fundamental breakthroughs in understanding neurological disorders like Alzheimer's disease or Parkinson's disease has come from analysis of postmortem brain tissue. In fact, for Alzheimer's disease, most of the therapies that are being researched now and even attempted are based on studies of postmortem brains from people that had Alzheimer's disease. And we realized, first and foremost, that autism and related neurodevelopmental disorders do not have a long history of study of postmortem brains like Alzheimer's. So in fact, the first studies were only done in the 1980s, which is relatively recently. And part of the problem for research in autism is that there was not an established resource of postmortem brains that could be distributed to researchers around the world who would have the tools to evaluate them. And so in 2013, we generated a proposal that we sent to the Simons Foundation Autism Research Initiative, and also Autism Speaks, who initially was one of the funders for Autism BrainNet. And they decided that this was a worthy cause and that it was something that they were willing to support. So we were formally launched in 2014 with the goal of building an essential resource for research causes of autism-related neurodevelopmental conditions. And what I want to emphasize is that even though we're looking at the brains, postmortem brains of departed people who had autism and neurodevelopmental disorders during life, our goal is to find targets for better interventions and treatments for those living with these disorders. So it really is looking towards finding targets for more beneficial treatment in the future for those who are with us. This is a collaborative project. We have 3 collection sites around the country. So we have one here at the UC Davis Mind Institute based in Sacramento, California. We have a second one in Boston at the Beth Israel Deaconess Medical Center. And then we have a third at UT Southwestern in Dallas, Texas. In addition to these 3 collection sites, we have international partners. In Canada, we have the Douglas Bell Canada Brain Bank, and we also have a partner in the United Kingdom at Oxford University that can acquire donations from Europe, from the UK and from Europe. And I want to emphasize that regardless of where a donation goes to any of these collection sites, we consider it one resource. And when an investigator is approved for receiving tissue, tissue can come from any of these collection sites. So we work really as a unified collaborative network to collect postmortem brains. You hear that some of the team, the team is actually much larger, because at each of the collection sites there are staff that are involved in making sure that the brains are collected properly, that they are processed and stored properly, and that they are safely stored for future distribution. But you can see, for example, within the Simons Foundation, the managing director is Dr. Marta Benedetti. You see Carolyn, but she has a colleague, Nicole Coleman, who is working on the clinical assessment of donors. And then there are Lilium, who's with us today, and Cece are both our outreach coordinators. And then we have both a big informatics enterprise, as well as we have a wonderful manager of the entire program, David Layden, who keeps it all functioning properly. So since 2014, we've had about 252 donations, which is actually a very substantial number. It's now the largest resource for autism research in the world. We've had 113 autism donations, and we've had additional donations. The big blue area here are control donations. Of course, when you try and understand what may be altered in a brain with autism or other neurodevelopmental disorder, you need to compare it with the brain of a non-autistic individual to see the differences. So we make every effort to also recruit appropriate control, we call them control brains or comparison brains. And we have a number of other brains. I won't go into the details here, but it's important for you to know in terms of Syngap that Safari has made the commitment to collecting donations from individuals who have genes that are implicated in autism, but even if the donor who had a variant of a gene implicated in autism did not have a diagnosis of autism, then we also can collect that donation as well. And there is some interest, of course, scientifically in trying to figure out why do some individuals, for example, with Syngap, have autism and others don't. We don't really understand that at the moment, and only by comparing those with and without the diagnosis can we come to that understanding. So why is brain tissue donation important? Well, the brain, of course, is the primary organ affected in neurodevelopmental conditions, including autism. We think by studying the brain that we will be able to develop either biomarkers or therapeutic targets. And as I mentioned earlier, brain tissue has contributed to understanding other neurological conditions such as Alzheimer's and Parkinson's disease. And we have every expectation that by studying the brains of those with neurodevelopmental disorders, we also will get insights that will lead to new and innovative targeted treatments. So you can study the brain in a variety of different ways. And we take a lot of effort in order to try, when a donation comes to us, to process the brain so that it can be used in as many possible ways, as many ways possible by a diverse set of researchers. So for example, you can look at the neurons or the cells of the brain. You can look for neuropathology. We can look for biomarkers in the brain that might be indicative of a disorder. There are many people that are utilizing the Autism BrainNet resource to look for genetic alterations. And here it's interesting that you would think, well, why do I need the brain? Why can't I just take a blood sample? Well, it turns out that there are different genetic alterations in different parts of the body. This is something that is relatively recently discovered, but there are somatic mutations that are different. So different cells of the body can actually have different modifications of DNA. And recent research using postmortem brains have actually shown that even different parts of the brain might have different genetic alterations. And so it's important to note that if you only look at the blood, you might actually be missing some of the really important genetic alterations that cause some of these disorders that we're interested in. And of course, the brain is a place that is affected by the environment. So there are other modifications of how the brain functions called epigenetic modifications that can actually be related to environmental contaminants or all kinds of other things. And this is a whole nother area of research that is going on nowadays that relies on postmortem brain tissue. I want to make the point too at this time that we, with the resource that we have, with the brains that have come in, we have an application process and researchers from all over the world can apply for tissue from Autism BrainNet. And if the application is approved, we can distribute tissue to them. There is no cost to the researchers for this tissue. It really is a resource provided by Safari to foster innovative and new research into neurodevelopmental disorders. I wanted to give you just one example of how things come together around postmortem research. I've been particularly interested in the issue of anxiety in autism. Turns out we've known even from the early days when autism was defined that anxiety is an important component, but we really haven't totally classified or diagnosed anxiety, particularly in individuals who have intellectual disability. This has been a big gap for many years. So we worked with Connor Kearns, who's an expert in diagnosing anxiety in autism, and carried out a research program that we published a couple of years back in 2019. And what Connor and our team found was that and I won't go into too much detail, but these are 11-year-old children that we've been studying since they were 2 in our program of research at the Mind Institute. What we found was in the typically developing children, the non-autistic children, about 8% of them had signs of anxiety. But there were 69% of the children with autism spectrum disorder who had an anxiety disorder, so a massive increase. And the question is why? What's going on with them? And we focused on a part of the brain called the amygdala. It's this outlined area here. It's a part of the brain that we've known for a long time is responsible for detecting danger signals in the environment. So in this little cartoon, for example, when this brain sees the snake, there's visual information that comes in. It ultimately gets to the amygdala, and the amygdala is the thing that makes you frightened and starts your heart beating, and you start your skin sweating, and hopefully allows you to escape the snake. Well, we've been studying the amygdala in autism using MRI for quite some time. And we found in this diagram here, it shows you that the size of the amygdala, this is the age of people that we have carried out magnetic resonance images of. And what you can see is there's a typical growth curve of the amygdala. Interestingly, the amygdala is one of the only parts of the brain that continues growing for a long time into adolescence and adulthood. And this is the growth curve for typically developing individuals. And what we find with our population of kids with autism is that there's a early sort of rapid growth. The amygdala actually gets too big too fast. But then when we look at older individuals, the amygdala is smaller. And we've wondered, what's going on? Why is this happening? And so Cindy Schumann, who's one of our node directors, she's actually the director of the collection site here at UC Davis, worked with a postdoc, Tommy Avino, to actually look at the amygdala in postmortem brain donations. And what they did was they used a fancy microscope to actually count the number of neurons in the amygdala of postmortem donations from individuals who had died at different ages. And what they found, and again, here's a chart that shows the age of the individual when they died and when the postmortem brain was donated. And the lines show you the number of neurons in the amygdala that they counted. The blue line is the typically developing individuals, and the red line is those who had autism during life. And what you can see is that early on, the amygdala of the individuals with autism actually had too many neurons. Yet over time, the number of neurons decreased, so that in adults, it was far lower than in the typically developing individuals. And this was actually the first indication that perhaps in autism, there is a degenerative process taking place, maybe not throughout the entire brain, but in the amygdala. And we think that this is interesting. We're trying to understand how this could then translate into anxiety, but it also has implications for other things that are going on in autism, like epilepsy, since the amygdala is one of the focuses of epileptic genesis in adults. So the bottom line is that not only do we find using MRI that the amygdala is too big early on and gets smaller later on, but we think that that's because there's actually an active process taking place where too many neurons are generated early on, and then for some reason they're decreasing, and it could be actually through a degenerative process that, again, we're studying even further now. You might say, well, okay, isn't it a good thing that there's too many neurons early on? More neurons, the better, right? But it turns out the brain is like a symphony. Everything has to be in tune. Everything has to work together. And if there's too many neurons early on, then probably means that connections are not being formed properly, and that the structure is probably not working properly as well. And it may be that the too many neurons early on produce an anxiety state that, due to that anxiety state, could lead to this degenerative process that takes place over time. Anyway, that's just one example. There are many, many other examples of how we're learning really new and interesting information from studying postmortem human brains. I'm actually going to let Carolyn start on this slide where she talks about our community partners, because she's very involved in that aspect of things as well. So Carolyn, do you want to take over?
Sure, happy to. David, that was an excellent synopsis, by the way. So we're going to shift gears here a little bit and talk, shifting from the science and things that examples of what we're starting to learn to a little bit more detail about how we operate as an organization. We wanted to first highlight our community partners because they are an example being, of course, the Syngap Research Fund. You are essential to what we do and integral to our ability to be successful in our endeavors. So our community partners involve medical examiners, organ procurement organizations, tissue banks, as well as family advocacy and research groups. And it's our hope that for our community partners, we're able to provide a very unique and important resource, in our brain banking initiative, and of course to provide support for your communities at a really important time if someone has passed away. We also do rely quite heavily on our community partners, and we rely on you to educate yourselves about Autism BrainNet so that you understand our inclusion criteria, so that you understand how a donation occurs and the process associated with that. And most importantly, we rely on our partners to know to refer families via our 24/7 hotline. And so actually we'll move a little bit more into the hotline and the donation process. So what happens when a family calls that 877 hotline that is available 24/7? The first thing that happens is they're connected with our answering service and indicate to the answering service that they're calling regarding a death and donation. And it's very important to use that language because the answering service is cued to connect you, and we can keep going, David, through the animation here. Because the answering service is then cued to connect the family or the hospital staff directly with our clinical team, who David introduced earlier, is comprised of myself and my clinical counterpart, Nicole. So once you're connected with Nicole, myself, or another member of Autism BrainNet staff, we work with the families and the hospital staff or whomever is involved to complete the informed consent or authorization for postmortem tissue donation process. We work with the families to figure out what is going to be the least burdensome ways to complete the authorization. Once that process is completed, we get to work sort of behind the scenes to coordinate the donation itself and to make sure that that donation is transported to one of the sites or the closest repository that David identified earlier. So it'll be Sacramento, of course, Dallas, or to Boston. So once the donation has arrived safely and has been preserved for research, Nicole or myself work to obtain as much information about our person as we possibly can. And that's so that researchers, as they're looking at the anatomical structure of the brain, have sort of clinical information to correlate with any potential differences, structural differences that they might see. And finally, we continue our work with our families really into the foreseeable future via our donor family aftercare program. And so just a little bit more about the donor family aftercare program. This is something that we find to be just an important part of what we do. We recognize the importance of what's happened to our donor families and want to honor their loved ones and provide our families with the appropriate support, as long as they need it. Participation in the donor family aftercare program is of course voluntary, but we want to, in addition to offering some mementos and support in writing, we also do want to stay engaged with our families via our Memories of Hope page. We send out a card every year to let the families know that we're thinking about them. And of course, we offer contact at any time. Family members can reach out to us via email, via phone, just to learn about what's happening with donation, but also many of our families want to engage in volunteer opportunities to support other families or to advocate for the idea of brain donation, and we welcome that as well. And finally, I think a lot of folks wonder, what is it like for a family to go through the donation process with us and to participate in an interview or to participate in the follow-up that I was describing earlier. And our experience has really been that our families appreciate so much the opportunity to talk about their person, and we very much enjoy learning about that person. So here's some quotes from our families. And last but not least, we'll leave you with our key points to remember, which include our 877, the 24/7 hotline, that we always follow up with you to provide with our families, of course, to provide bereavement support and the clinical follow-up that I was describing. And so this is something that we haven't touched on yet, but for families who are interested in long-term care planning, so in the same way that you are doing your long-term financial or medical directives, you can also consider brain donation now, and we will provide you with the materials that you may find helpful to include in those long-term care planning endeavors. And last but not least, a point that we always want to make is that there's no cost to families when donating to Autism BrainNet. And also there is no cost to researchers who are looking to utilize this very important and precious resource. And I think that's it.
Yeah, so we have more information, excuse me, we have more information on our website. Please feel free to go to our website. We also have a quarterly newsletter that Lilium is involved in developing. You can sign up for that at autismbrainnet.org/newsletter. It brings to you stories not only about the staff of Autism BrainNet, but also about donors and then some of the scientific advancements that have taken place due to postmortem autism research. We always are happy to answer questions. So you can go to our website as well and post a question to us, which we'll respond to as soon as possible. So with that, we'll open it up to questions. I'm going to stop sharing, and thanks again for your attention.
Thank you. Marcia, did you want to say anything about the anxiety?
Yes, I have a few questions. That was amazing, the part of the amygdala and the anxiety. And we actually, that put me to work right now, because I was thinking most of our patients' MRIs, they are normal. But I don't think nobody has dedicated to compare really the size of the amygdala.
Yeah.
That was a very amazing point, and it all comes down to, they look like by your slide, the peak age was after between 5 and 10 when the amygdala plateau, and then the number of cells goes down.
Yeah. Yes. So it actually becomes even more interesting and complicated, but I'll try and summarize it briefly, and that is that there's sort of standard anxiety that that people without autism or other neurodevelopmental disorders have. But then there's a form of anxiety that we call distinct anxiety that is really specific to autism and other neurodevelopmental disorders. And it turns out there's a paper that will be coming out, or it just came out actually, in Biological Psychiatry, a journal that is, well-respected journal in psychiatric disorders, that the amygdala shows a different pattern of alteration, whether the person with autism has the regular form of anxiety or this distinct form of anxiety. But really it points to the idea that the amygdala is the heart of probably the problem related to the genesis of anxiety. And Marta, it's a good point. When, if you look at the brain of an individual with autism, it doesn't look different. It really necessitates some very detailed and sophisticated analytic techniques in order to get to the point where you understand that something like the amygdala is altered. And it's not altered in the same way in all people with a particular disorder. So you really have to have, number one, a big population, and then you have to use these more sophisticated analytic techniques. But when you do that, then you start actually finding some interesting differences. And of course, we'd like to know what causes those differences and whether they can be treated. Again, this is always our goal. Do we get information that allows us to do something that's going to help people with these disorders?
Yeah, another thing I pick up from what you said was we have a big amount of kids with normal EEGs that we know they are having seizures. And we always think it's located in the deep brain. And of course, the amygdala is a very deep brain. And that was another thing. It was quite interesting that you guys find out that it's a very good location for seizures in adults. And that was very interesting to know.
Thank you. I think, for something for the foundation to think about down the line is maybe to mobilize, an MRI analysis of the brains, trying to get a larger group of individuals with Syngap, and then focusing on some of these more innovative techniques for analyzing the brain. Because again, a standard sort of clinical scan, you may or may not see anything, but I think once you actually start using the techniques that we and others, we're not the only group that does it, but there are, I think, very sophisticated techniques. You just need larger numbers of cases, and that's the point, to be able to make sense of this. But yeah, I find, in autism sort of generally, there are up to 30% of individuals that will ultimately have a seizure disorder. And the data says that 50% will show a seizure early before puberty, but 50% of them will have a seizure after puberty into the late teen years. In fact, I mentioned, the Mind Institute was started by families who have children with autism. Those children now, we're about 22 years that we've been doing the Mind Institute. Those children are now adults. And, 2 or 3 of them actually had their first seizure when they were older teenagers. And, the question is there anything that we can understand about the development of their brain that's going to predict that that's going to happen? Maybe there's a prophylactic treatment that we could, introduce in order to try and avoid. And again, it's a sad situation, but many of the donors that we receive at Autism BrainNet are individuals who have their first seizure and actually die from their first seizure. And again, it's a situation where hopefully the research will get to the point in time where we can predict who's going to have a seizure earlier on and avoid that devastating event in the family's life. So thank you for those comments, Marta. I'm glad that there was some synergy in what we said and your interests.
There's huge synergy. I want to make a couple comments, if you'll permit me. The first is, once our families get seizures under control, the next thing I encourage them to do is talk to their pediatrician about anxiety meds. So I do see that anxiety is prevalent in our population and leads to a lot of behaviors and challenges for everybody. And I took a screenshot of the slide with the anxiety stats, so I'm going to use that. But I want to bring it back to brain donation for a second, and I want to share with people how I found you. And after I talk, I'm going to invite Nancy to share. But we have, so to be clear, for any newly diagnosed families who are watching this, because I know there's some people on Facebook watching, Syngap is not neurodegenerative. Our kids do not die of this disease. And to my knowledge, no Syngap patient has died of seizures, although SUDEP is something we should all be mindful of.
Right.
With that said, we do have a few older patients, and Nancy, who's the sibling of Karen, who's the oldest living patient, called me and said, hey, I want to know what to do when Karen passes. I want to make sure that, we take full advantage of the life she's led and what she'll leave behind. And so I reached out to our scientific advisory board, some very prominent people like yourself, and said, where do I go to donate a brain? What do I tell this family who's asked me this important question? And I got put on the phone with a lot of important people who run brain banks. And it was very much like, I got to them very fast. And then they were like, okay, who's dying? Where are they? And I was like, no, they're not dead yet, but I just want to plan. And they were like, oh, call me when they're dying. And it felt very, they're very busy people running important programs, but it felt very transactional. And I thought to myself, this is not what I want my families to go through. And then, I love that you put up that partner page, Carolyn, because as I was looking around, we reached out to other rare disease groups who I respect tremendously. And you had a lot of them on there, Angelman, FOXG1, KIF1A, Phelan McDermott. And I said, what are you guys doing? And again, I heard Autism BrainNet. And I said, oh, let me talk to these guys. And two things struck me. One, you're part of the Simons Foundation, who I know and respect and does everything well and is very well funded. So cost was not an issue. And two, there were these very kind, good people who were there to talk to and support families. And that struck me as a huge service. And as somebody who leads this organization, I didn't want to go back to Nancy, who I respect so much, and say, you should work with them unless I was sure. And between the endorsement of other patient organizations and the great experience I had talking to you guys, I was very comfortable. And then we signed an agreement that said, if you guys get some Syngap tissue, as people request that tissue, you will let us know about that request and ask our opinion. We don't have any authority. We don't get to make any decisions, but we get to say, oh yeah, great guy, or never heard of this one, or maybe we should talk to him, whatever. And I was so grateful for that post-donation connectivity, because while our children don't die of these diseases, if our children die from seizures, from COVID, from whatever, it's such a rich resource. To my knowledge, there's like one Syngap brain that's been studied in the Vlaskamp paper in 2019, but it's not sitting in the States. It's not available. So I just want to emphasize to people that we didn't just find you guys and sign up on the website, right? There was a huge process I went through. And I really, since I'm willing to stand before my community and say, I think Autism BrainNet offers a valuable service. And when, God forbid, any of us have to go through this, I feel comfortable that you guys do a great job supporting them. So with that said, if I've left you anything to say, Nancy, can I bring you up and ask you to share your thoughts? Because you have had the sort of pre-meeting, right? Are you there, Nancy?
Here she comes.
Here she comes. And I think most people know, but Nancy's on our board and she's Karen's sister. And Karen is the oldest living Syngapian. And
okay.
Thank God.
Yeah,
Hello.
nice to meet you. Yeah, hi, Nancy.
Hi. Hello, everyone. It's so nice to meet everyone. So it was I was thinking Karen was very ill and then she got better and survived. And I kept thinking on my license, I have that I'm donating my body parts if something happens to me. And I was thinking I'd like to do the same for Karen. I found out she had Syngap and I wanted her legacy to continue. I wanted Karen's life to mean something and I wanted to make her to make a difference. And I just wanted to continue. So I reached out to Mike. Mike did all of this wonderful work and came back to me and shared the information about autismbrain.net. And then I received information from Carolyn Hare, and Carolyn and I spent a lot of time talking, and she was just so wonderful and calm and explained everything to me. And I spoke to my other sister, Jennifer, who is co-guardian, and she's like, oh, absolutely. She said, I have that on my license too. We should, absolutely be thinking along those terms for Karen. So we completed, some of the paperwork and got things moving along, and I just have peace of mind now. It's just, it's on the back burner, and Karen's turning 66 soon in 3 months. So she's healthy. That's great. But it's just on the back burner and peace of mind. And I feel really good about this decision, really good about it. Even better now after watching this webcast, webinar.
Yeah, thank you, Nancy. Mike, I just want to answer a question that Heather put in the chat box, just because it's a really important question. So the question was, since the brain isn't removed during embalming, can it be donated after the funeral, or does it have to be donated immediately after death like other organs? That is an absolutely critical question. And we make every effort, and we would ask that that we be called as soon after death as possible. And I'll just say that the usefulness of the brain for scientific research depends on the time after death in which it's acquired. So the brain will change over time. We want to see the brain the way it was most like when it was in the living person. And so the answer is, just like an organ that you would donate for transplant that is most beneficial quickly after death, the brain donation has to take place as soon as possible after death. And so we're talking about, 24 hours is 24 hours or less is optimal. We will accept donations longer than that. But some people think, well, it doesn't really matter if we have the funeral and go on and on and on. The longer the brain is not donated, the less it's going to be useful for research. So just keep that in mind when people are thinking about making a donation. And I'm glad, Nancy, that you've had such a good experience, and I'll second the note that Carolyn is just absolutely wonderful, and she can never retire from this. Because she would not, she's irreplaceable, and we need to keep her doing what she does so well.
Dr. Amaral, if you can go, we have a question from Hans on the Q&A, and we have a hi from India from a PhD master that is doing actually MRIs, studying MRIs, and I think he's very interested in this.
Yeah, I'm looking at it. Okay.
Hans' question.
Yeah. Yeah, that's terrific. So he's doing research on children less than 3 years old with and without autism. That's really, really important. And in fact, at the Mind Institute, we also are doing research starting with kids when they're first diagnosed at 2 to 3, and then following the development of their brain over time. That's the only way that we could actually see those changes in the amygdala over time. So I would encourage you to keep going. If you want to talk more about your research or let me know what you're doing, just send me an email and we can talk offline. I'd love to hear more about what you're doing. In the Q&A, let's see the question. To get our Hans, I'm sorry, I was looking in the chat, so thank you. To get our diagnosis, my family did trio testing. So as a dad, I've registered not only my son, but also us for trio brain donation. Oh, fantastic. That way researchers can have genetically related samples with the age cutoff that seems unavailable with ABM. Am I mistaken? No, the age cutoff really applies to control donors, but in the case that you're talking about, Hans, where it's a family member, we see that as very valuable. And again, it's many cases are idiosyncratic, and we want to work with families. So in your case, if you're interested in the possibility of doing a trio donation, please contact us. We can make special arrangements for that to happen. And we do get asked by our scientific researchers if we have parents of children with autism available in our collection. We have a small number, but I don't think we actually have a trio yet. And so we would welcome you to contact us and see if we can arrange that in a long time from now. Thank you, Hans, for your question, though.
Another question, while somebody else go. For example, in Syngap, we know that our children have a slow GI tract, and you only get the brains, but what happened with the rest of the tissue? I wonder how many people has that question with the rest of the body.
Great question.
For
Yeah.
example, for us, it's important GI, to put one
Yeah.
example.
Yeah, no, that's a really good question too. And we've been working on that question. We haven't come to a final decision about how to work this out. We've had a couple of cases, not with Syngap, but with other genetic disorders where other body organs are really affected. So we had one case where the kidneys and liver and peripheral nerves were actually affected. We knew ahead of time that researchers would be interested in those organs. So we made a special situation where we actually did collect those as well. The GI tract is really interest, I think, across autism. And we're going to have our next sort of policy meeting in October of this year. And I think one of the big questions are we going to go to collecting GI, routinely? You can think about it, every time we make a decision like that, it has costs associated with it. It has, lots of logistical kinds of things. And so we wanted to make sure that the brain being the most important organ, that we had a good system for doing brain collection. We feel like we have the optimal system now. In fact, and I wanted to mention this too, the other collection in the United States for autism and neurodevelopmental disorders is the NIH Neurobiobank. We don't compete with them. We collaborate with them. And in fact, we recommend that when researchers want tissue, that they look at both sources. And in fact, we have an ongoing communication with people at the NIH Neurobiobank, and they've now agreed to process their brains that are donated with individuals with autism in exactly the same way that we process the brains, which we think is a little bit more sophisticated than the way brains have typically been processed. And in order to make sure that regardless of which place you get tissue, it's going to be the same quality and type of tissue. So we will probably have a decision on how commonly we'll collect GI tissue. My guess is that we're probably going to try and figure out a way to do it, because I know it's actually the GI system has as many neurons, or close to as many neurons as major portions of the brain. So it's sort of the second brain. And again, GI problems affect huge numbers of people with these disorders, and we don't understand them either.
Yeah, Syngap is the same. So again, bring it back to our community. Most of our parents are more terrified of dying before their kids than burying their kids, because we're worried about who's going to take care of them and all the issues they're in. But if a parent has, if there's a sibling like Nancy who's got needs to be mindful of this stuff, or if a parent has a kid who is sick and can see around the corner, I don't know, Carolyn, can you just talk for a little bit about the value of doing a little bit of pre-work in terms of just what can be done in advance to reduce the burden in the really difficult time when someone passes?
Yeah, absolutely. And it isn't, it's we're very aware of the fact that this is a sensitive conversation to have, but at the same time, it is helpful for families like Nancy to take some initiative in reaching out. Having the ability to plan a little in advance is always very helpful. Number one, we can understand more about the person we're talking about. We can do some of the logistics, the literal coordination of donation ahead of time by connecting with all the right people. We can provide our families with all of the forms that they'll need at the time of death. And I think of equal importance, and as a follow-up to the conversation that we were just having, if there are special considerations like there are with Syngap, with the GI tract, we would want to understand that so that we can make the extra effort to accommodate those specific needs and interests.
Great, thank you.
Yeah, of course.
Nancy, you going to say something? Anyone else have questions?
I just want to point out that another thing I catch on that was that a lot of our Syngapians don't get the diagnosis of autism early, and or don't get never the autism diagnosis. And I'm glad the rules kind of changed lately, that is more inclusive. But I'm glad that you guys, even just with the genetic diagnosis, will take the sample. It doesn't have to have autism diagnosis.
Right.
I guess we have an atypical autism. Our kids are very cheerful and yeah.
Well, and again, this is getting more complicated in the sense that we're using our inclusion criteria as anybody that has a variant in any of the spark genes. So the spark genes, I think now there's 153 genes that are implicated in autism and related disorders, and something like 16 copy number variations. So probably most people don't know that they have one of these variants, but in the next decade, I think more and more people will have, next generation sequencing, and they will actually find out that they have, Syngap or something else that is related to the intellectual disability. And the Simons Foundation, again, is very keen on understanding how variants in the genetic architecture of individuals can lead to some of these neurodevelopmental disorders. So yeah, and Mike, going back to what you said before, you did an incredible due diligence. I felt like we were sort of on trial, but that's fine. And I think it's been a pleasure for me to work with the Simons Foundation, because what you said is absolutely true. They, Jim and Marilyn Simons, who set it up from the very beginning, wanted to do, number one, it was totally altruistic. They wanted to help people, and they wanted to help, the autistic and other neurodevelopmental communities. And number two, Jim's a brilliant guy and Marilyn's a brilliant woman, and they wanted to make sure that their organization reflects brilliance as well. And we try and live up to their example. It's sometimes not easy, but we try and live up to their example. So we're glad you put in all the effort to select Autism BrainNet to partner with, and we work hard to be worthy of your trust.
Absolutely.
Thank you for saying that. It's very nice of you. I appreciate it. I'm sorry I made you feel like you were on trial.
In the best possible way.
I just didn't.
In the best possible way. I'm just kidding.
If there's no more questions, we're going to wrap up on time, which is great. I really appreciate this, and hope that this presentation helps families think about this as and when they're ready.
Yeah. Thanks again, Mike, and everybody. We appreciate it.
Thank you so much.
Okay.
Thank you for having us.
Yeah.
Thank
Take
you
care.
so much. Thank you.